Morning, everybody.
Morning.
Today, we're going to be on 18.2 today, talking more about viruses and what they do. So today we'll look at the different kinds of viruses, the different structures and shapes they have, and how they attack different cells. Key concept is that viruses exist in a variety of shapes and sizes. So viruses differ in shape and in the ways of entering host cells. These, you can see different kinds of viruses and how different shapes they can be. You can have a spherical shape. That's influenza virus. It causes the flu. A complex shape like this one attacks bacteria. You can have a helical shape, and that infects plants. Polyhedral shape. So these different shapes allow them to enter the cells in different ways. This complex virus right here has a needle that injects its DNA into the cell. So the virus itself won't enter the cell, it injects DNA into it. So one like this, though, that's circular, it will actually be absorbed into the cell. And so some are designed to be absorbed in, like a circular helical one. Others are designed to inject the DNA into the cell, but staying on the outside of the cell. Viruses have a simple structure. There's only a few parts to them. So this one right here, you can see that it has RNA on the inside. Either RNA or DNA will be on the inside of the virus. They won't have both. It'll be one or the other. And then there's a capsid. It's a protein structure that protects the virus, protects the DNA or RNA inside the virus. So the most important thing that viruses have are genetic material. It has DNA or RNA. And then DNA or RNA, there's a capsid, and it's a protein bubble that protects the DNA or RNA. Now, some also have a lipid envelope, and that's on the outside. So beyond the capsid, you might have another layer of protection, and it gives it more protection. It also might have surface proteins that are on the lipid envelope. We'll talk more about what those do later on, but it's kind of like the key to get inside the cell. So these surface proteins act like keys that allow a virus entry into cells. So this shows you just different kinds of viruses and the parts of the virus. They all have a nucleic acid on the very inside. Usually it's wrapped around in a spiral shape. It can be DNA or RNA. then you have the capsid that protects it, and then you might have a lipid envelope with surface proteins on the outside. So this one shows you the flu virus, this one causes rabies, and this one causes foot and mouth disease. This kind of virus is called a bacteriophage, and a bacteriophage infects bacteria. PHAG, phage, means eating, and so this name literally means bacteria eater. And so it has a DNA up in the capsid right here, and when it latches onto a cell, it's going to inject these needles will pierce the cell's membrane and inject DNA into that cell. Okay, don't worry about this one. You already wrote that one down. Just write down bacteriophage infects bacteria, which I think you already did, but just in case. So this is just another view of what a bacteriophage does. So it will latch onto a bacteria. That's what this cylinder-shaped structure is. And it's going to, you see little needle parts right there. That's where it pierces the membrane. The DNA is up here, and the DNA injects into the cell. And this is just a close-up view of it. So this is a microscope image of the viruses attaching onto the bacteria, and then another microscopic view of the bacteria too. So they're all in green on here. The viruses are, and the bacteria is in brown. So bacteriophages pierce through bacterial cell walls, injecting DNA into the bacteria. There's another way to see what happens with the bacteriophage. So you can see the DNA goes into the cell, that's in red right here. Once the DNA is inside the cell, here's instructions to make more viruses, and it'll start to make more viruses within the cell. And eventually the cell's going to burst and send out the viruses from that cell to infect other cells. And we'll talk more about this a little bit. So don't worry about writing this down at this point, the image anyway. We'll talk more about this process later on. So that was for prokaryotic cells, bacteria. For eukaryotic cells, the viruses actually will enter. And so cells that are found in plants, animals, and humans, the viruses actually won't stay outside. They're going to enter into the cell. And that's what we see in this picture right here. So the virus is the circular green thing. This is a cell, and the cell is going to absorb the virus into it. Once inside, the DNA from the virus, you see in gold right here, will create more viruses. So endocytosis, we might have mentioned this earlier, but it means taking things into the cell. So endocytosis is the process that you're seeing right here, where the cell membrane absorbs something from the outside and brings it into the cell. And this is just another way to see the process where a virus enters the cell. So the cell membrane brings it in, then it starts to create smaller viruses, which might then leave the cell and come out and infect other cells. And we'll talk more about this process too later, so don't worry about writing down this diagram right here. We'll talk more about this in detail as we go on. Just know the main idea that eukaryotic cells bring viruses into the membrane. So this is important to know, this diagram on here. So viruses cause two kinds of infections, and the first kind is called a lytic infection. In a lytic infection, eventually the host cell will burst with viruses. So it starts up here in the very top right, where a bacteriophage injects DNA into the bacteria. And then the DNA is in blue on here. So this blue DNA is what the virus injected into the bacteria. That is instructions to make more viruses. So now that that DNA is in the bacteria, more viruses are going to be made, and they're going to overwhelm the cell. Eventually, as the viruses build up, the cell can't hold them all, and the cell bursts, and viruses emerge from the cell to infect other bacteria cells.
Do you want us to write this down, like to draw it in a notebook?
Yeah, it'd be good for you to know this one, to know this diagram. Um, so I definitely encourage you to draw out this diagram in the process, yes. Now, this only takes place in bacteria cells. So the lytic infection doesn't take place in humans, animals, or plants. Those are all eukaryotic cells. Lytic infections only take place in prokaryotic cells and bacteria. So this is one of two things that can happen when a virus infects a bacterial cell. We'll talk about the other one a little bit, but this is the first thing that could happen when a virus attacks the cell. And this is of the two possibilities, this is the more dangerous one for the cell. This results in the cell bursting and dying. So this would be an active virus causing damage to the cells. Does everybody have this down?
Almost.
Okay. All right, so that's the lytic infection pathway. The other thing that can happen when a virus infects the cell is called the lysogenic infection. So this one would cause no immediate harm to the bacteria cells. It's more of a dormant form of the virus. So in both cases, though, you have a bacteriophage that injects DNA into the bacteria. And so you see it's in teal right here. The DNA becomes part of the cell's DNA. So magenta represents the cell's DNA, then the teal represents the virus's DNA, and it becomes part of the cell's DNA now. And so that means every time this bacteria cell divides, it will also copy the virus's DNA along with it. And so as over time, you'll have more and more cells that carry the virus's DNA. Now, it might be inactive for some time, which means the cells are healthy. But if they're under stress, if things change in the cell, then that DNA can become active and create viruses that will then start to destroy the cells. So this is just the same thing that I showed you in the last slide. It's just more steps now. So I showed you the first 3 steps on the other slide. But what can happen after step 3 is if the cell becomes stressed, then the virus's DNA can come out and become active. And if it separates from the bacteria's DNA, it will then start to make more viruses. And this is now the lytic cycle that we saw in the last slide. So it can go from lysogenic, where it stays dormant and the virus's DNA just stays with the cell's DNA, to the lytic cycle, where the viruses start to become active and start to be made, at which point they'll overwhelm the cell, they'll burst the cell, and then they'll infect other cells when they come out.
So do you want us to draw this one too?
Yes, this would be good for you to know the steps. Yeah, so you should draw this as well.
Okay.
So these top 4 steps, that's the lysogenic cycle. The bottom 3 is the lytic cycle that we saw from the last slide.
Could you repeat that?
Yes, so these top 4 steps right here, that's the lysogenic infection. So that's when the virus will inject the DNA into the cell, but it's not active, it stays dormant. These bottom 3 steps right here, that's the lytic infection that we saw on the previous slides. That's when the virus becomes active, when it starts to create more of its own kind, and it eventually will burst the cell and send out more viruses in the process. So these two are connected. Lysogenic can become lytic, lytic can become lysogenic. So these can go back and forth between the 2 cycles. If the cell is healthy and not stressed, it might stay in lysogenic, where the virus is not active. If the cell is stressed, it can go to the lytic cycle, where the virus becomes active and destroys the cells.
Which one would be more dangerous, though?
The lytic cycle by far. That's when the virus starts to destroy the cells. As long as it stays in lysogenic, it's not going to hurt the cells. Does everybody have these diagrams down? Okay. And this shows you just another view of how the cycles can go back and forth from each other. So you can see right here the lysogenic cycle up here in this top half. So that's when the virus will inject DNA in. You can see it's in red right here. The bacteria cells will divide, carrying the viral DNA. It'll keep on duplicating itself. It can then enter the lytic cycle, where the viruses start to be made within the cells. And then over time, they'll destroy the cells in the process of producing more viruses within the cells. You don't need to draw this one down because you already drew it on the previous slide. So this just shows you how they're connected to each other, but you don't have to draw this diagram down. Are there any questions about the two cycles, or do you need me to go back to any slides? Okay, well, I want to show you a short video before we end class today. This shows you more about what we talked about today, about how viruses attack cells, how they enter cells. Let me pull it up for you all.
Miss Holly.
Yes, go ahead.
For number 4, would it just be lysomic because it's not like a disease? And it's not harmful?
So you want to choose one or the other between lytic and lysogenic. Which one do you think it would be?
Lysogenic.
Exactly. Yeah. But I couldn't hear your full question. What was your question about it?
Oh, just that, like if it would be lysogenic because it doesn't cause any harm.
Oh, yeah, you're right. Yeah, I misheard your question. Yeah, that's correct.
Thank you.
Miss Holly? For number 5, how would you answer the question using natural selection?
Okay, so for that one, it's asking, how is it possible for the proteins of its capsid to match receptors on the host surface? Well, if it doesn't have the right receptors, it won't infect the cell. If it won't infect the cell, it can't survive and pass on its traits to other cells. So it would only be the viruses that can survive and attach to the cell and pass on their traits to future cells. So the ones that don't have the receptors will just die off, and the ones that have the receptors will survive and pass it on to other cells.
Okay, thank you.
You're welcome.
Thank you.
Welcome. Have a good day.
Thank you.
Welcome. Have a good day.
For number 3, researchers studying infection can often grow bacteria, blah, blah. What conditions does scientists provide for viruses to multiply? I'm looking in the book, but I think we talked over this, but I'm just looking in the book and I can't find it.
Well, think about why viruses aren't living things. Do you remember why we say they're not alive?
Because they can't reproduce on their own.
Exactly. So what do they need to reproduce?
Oh, a cell.
Yeah. So that would be what you would say for that one.
Oh, okay. Thank you.